All right, so now that we've covered the cell cycle and its regulation in the previous lecture, in this third and final lecture, we're going to talk about clinical pearls relevant to the cell cycle, and that will include a quick discussion of cancer biology as well. So first, we're going to talk about cancer biology. Cancer is a subset of diseases that are fundamentally a result of dysregulation of cell growth. And there's two categories of genes that are affected that can cause cancer, and those are oncogenes and tumor suppressor genes. Now, we've talked about tumor suppressor genes a little bit already. So first, let's talk about oncogenes. These are genes that have the potential to cause cancer. So they're not necessarily just always going to cause cancer. They have the potential to cause cancer. And they usually code for proteins that promote cell growth, reproduction, and/or signal transduction. And that makes sense. These are processes that are intricately involved in cell division and DNA replication. And so it makes sense that these are the ones that would be affected. And usually they code for receptors. So we'll draw our little cell here. So you have a receptor. Then a receptor, when it gets activated, it's going to activate a signaling protein. So signaling protein. And then you can also have, in the nucleus here, you can have transcription factors that can either be tied to a signaling or they act on their own. And so then they bind DNA and then induce their own transcription changes. And so oncogenes, they typically, again, they will code for one of these types of proteins. Next, you have a proto-oncogene, which is a normal gene. The key thing here is a normal gene, because remember, these are all normal proteins. So the genes for these are all normal. And this could become an oncogene as a result of either a mutation, induced overexpression, or chromosomal translocation. So now let's talk about that over here. So first, here's your gene right here. This is your proto-oncogene, so it's a normal gene at first. And then let's say it has some kind of mutation that occurs. Either it's inherited, or let's say it gets exposed to some kind of damage from a carcinogen, such as UV light, or from cigarette smoke, or from nitrosamines, or some other type of carcinogen. So the gene gets damaged, gets mutated. It results in a dysfunctional protein, either a protein that cannot be regulated or a protein that does not function at all. An example, you could have a receptor that can't be regulated, so it's turned on all the time. It can never be turned off. An example of that is when you have EGFR mutations, and these are involved in a lot of cancers. It's turned on all the time. So it constantly promotes cell growth, cell proliferation, cell division, and so it causes uncontrolled tumor development. And so obviously that's why then it causes cancer. And so because it just can't be regulated, and because a receptor, you don't want it on all the time. You just want it on when it's activated by the specific ligand. Versus if, in this case, it's just active all the time, even when the ligand's not even there. All right, so we'll clean the slide here and we'll show you what induced overexpression is. So we've got our gene here. So you can have increase in expression of this gene through just misregulation. So either the transcription factor that's supposed to turn it off is mutated, or there's a transcription factor that turns it on that's mutated and it's constitutively active. And so you're just constantly overexpressing it. That's one mechanism that can occur. You can also, so remember, these genes, this is going to be your DNA. You're going to produce an mRNA for this gene. It gets translated into protein, and then it gets degraded. However, there's situations where this doesn't get degraded, and the mRNA is still around and can still be expressed into protein, which is going to dramatically increase the levels of that protein. So again, that's another way you can overexpress a proto-oncogene and then turn it into an oncogene where it's causing cancer. And then you can have essentially what's called gene duplication, which is a chromosome abnormality. We won't get too much into that, but essentially it's in the name, you get two of these genes. And so as a result, as having two of them, you're going to have twice the mRNA and twice the amount of protein. And so again, much more of that protein than you want. And then third, you have chromosomal translocation. We'll talk about this when we talk about CML, or chronic myelogenous leukemia, a famous example with the drug that's involved in that, which is imatinib. So chromosomal translocation is when you essentially you can take one piece of a gene move it on to another chromosome, and then you put it next to your gene. Let's say you have gene 1 here, which got translocated from another chromosome, and then you put it next to gene 2 here. That results in overexpression. So because of this, let's say it's tied to a promoter that's not as tightly regulated as gene 1, and so then it gets overexpressed. That could be one example. Another example is where, let's say you have, and this is what happens in CML, is you have gene 1 that gets fused with gene 2. So you essentially create a new gene, then you create a fused protein. So you have your mRNA. So you create a new mRNA, and then you create a fused protein. And this protein, as in the case of CML, is then not subject to either regulation, is constantly active, and then can also lead to cancer. So I've gone through what each of these mean. The bottom line here is that you have a normal gene, because all of these are normal proteins. At the end of the day, they're all normal proteins. It's just that they have modifications that happen to them through DNA damage or mutations or any of these other mechanisms I've talked about, and that's what then can cause cancer. So you have a normal gene. These are, remember, proto-oncogenes, because remember, a proto-oncogene is a normal gene. So this is the very early in the process. Then you have DNA damage, which again can be from carcinogens, or you can have a mutation, or both. Then you have an oncogene, which again, you still haven't gotten to cancer yet. It has the potential to cause cancer. Then let's say over time you develop even more overexpression of this oncogene. That's what will give you cancer. Tumor suppressor genes, these are genes, as we've talked about, that code for proteins that inhibit cell division and or promote apoptosis. And if you remember, p53 is a great example of a tumor suppressor because it actually does both of these. The other thing that tumor suppressors do is they ensure that DNA damage is coupled to the cell cycle. Okay, because remember, then you want to halt the cell cycle and then go back and repair the DNA damage. That's the other thing that tumor suppressor genes are involved in, is promoting DNA repair. So either remember, p53 helps induce transcription of DNA repair proteins. Another example. So they're all about regulating cell cycle progression, preventing DNA damage or repairing it, and making sure that the cell cycle doesn't progress in an uncontrolled manner. It's keeping things nice in check and nice in control. The other thing is, let's say the DNA damage is beyond repair. Okay? And remember, p53 is involved in this as well. It's beyond repair. Okay? Then you want to induce apoptosis. And you hate to see the cell go, but the problem is if it's got too much damage, it's mutated, you don't want to pass that on to subsequent cells because you don't want to get cancer and you don't want to have uncontrolled cell growth and tumor generation. So you induce apoptosis, you just cut your losses, move on, plenty of other cells to go around. Now, real quick here, we want to talk about some chemotherapy drugs or anti-cancer drugs that target specific phases of the cell cycle. This is really high yield for biochemistry exams, really high yield for board exams. Really almost a guarantee you're going to see one of these on a board exam. So the way it's going to work is they're going to give you a drug. They're probably going to be talking about giving it to a patient that has some type of cancer. And they're going to have this big long stem. And at the end of it, they're really going to ask is, what phase of the cell cycle is this drug acting on? And so you want to know that. That's why it's important to know what's going on in each of these phases, because then you can know where you can think about the drug mechanism and then apply it to that, and then you can know where it's acting and you can answer the question. Again, biochemistry at first, it seems like there's a lot of memorization involved, and there is a decent amount, but the other thing is you want to really understand things as much as possible. When you're learning these biochem, anatomy, physiology, pathology, you want to always be asking why. Why do these things happen? And if you can answer that and tie these things together like we're going to do here, it can make it so much easier to remember it. And then it also just makes it more applicable because it's, at the end of the day, you're learning this to some degree to apply it to patient care. So first, anti-metabolites. And we talked a lot about this in the first lecture. So you have methotrexate, 5-fluorouracil, and hydroxyurea. These inhibit purine pyrimidine synthesis. Remember, we talked about that all the way back in the first lecture. So you're essentially inhibiting DNA synthesis. You know that these act on those pathways. You know that they act on DNA synthesis. So if the question's asking where in the cell cycle, you just got to think about where does DNA synthesis happen? Well, here you go, S phase. So these guys all act on the S phase. So it doesn't matter any of these three drugs. Sure, they each have their own mechanism, but in the case of this question, they all act on the same phase. They're all having the same general end result of inhibiting DNA synthesis. And so that's going to be the S phase. So topoisomerase inhibitors, two examples here are etoposide and irinotecan. Etoposide, we'll start with that first. This acts on topoisomerase 2, so it inhibits that. And remember, topoisomerase is involved in unwinding DNA and then helping prevent strain in DNA. So it actually induces some breaks, as we talked about in the DNA. It allows it to kind of untie itself, if you will, and then you have to religate those strands that you broke to put it back together. And by doing this drug etoposide specifically, by blocking topoisomerase 2, it prevents that religation of the broken strands. So then it increases the amount of DNA strand breaks. And so as a result of that, during G2, you've replicated the DNA. You want to make sure everything's ready to go for mitosis. So you want to do some unwinding. You're also repairing DNA damage. But as far as topoisomerase, you're unwinding strain in the DNA so that it can be properly distributed to the daughter cells. Now, the problem is if you can't religate that broken DNA, you're going to have problems. And so what's going to happen is that the cell's actually going to detect all those breaks and it's going to induce apoptosis. Now, what's interesting is cancer cells rely on topoisomerase much more than regular cells because they're dividing so much more. They're going through the cell cycle at such a faster rate. So this is really going to affect rapidly dividing cancer cells. Long story short, it acts in the G2 phase, which makes sense because it's after you've replicated DNA and you're getting it ready for mitosis. And as a result of blocking this, you're going to induce DNA degradation as well. Now, irinotecan, this prevents DNA unwinding because it's going to act on topoisomerase 1. And we talked about this enzyme in the DNA replication lecture. And so that's during DNA replication, that's going to be during DNA synthesis. Remember, this helps prevent that strain. It helps decrease the strain during replication. DNA replication to be exact. So that's what topoisomerase 1 does, if you recall. And so DNA replication, this is right in the middle of the S phase. This is what the S phase is. And so as a result of that, you're going to block DNA replication because a big part of DNA replication occurring is preventing this strain and decreasing it. And so you're going to, again, affect rapidly dividing cells that are replicating DNA at a fast rate, which are going to be cancer cells. So bottom line, etoposide is G2. Irinotecan is the S phase. Then you have bleomycin, which is an antibiotic, and this induces free radical formation that leads to DNA strand breaks. So it interferes again with DNA repair. So again, during this phase, you're going to have DNA repair because it's right after you've replicated the DNA. So thus it's going to act in the G2 phase. So microtubule inhibitors, you got paclitaxel, vinblastine, vincristine. The simple answer for all three of these is they all act on the M phase because they're all acting in mitosis. And as you saw in the previous lecture, you saw how much microtubules play a critical role in mitosis and cell division. So if you get asked what phase these act on, it's going to be the M phase. However, you also, just to review this, you can have really two mechanisms here. Either they prevent mitotic spindle breakdown or they inhibit mitotic spindle formation. So breakdown, if you remember, you have metaphase. And where there's a few areas where this can affect, but one main way is where during metaphase you've got the chromosomes that are aligning like this, across the center of the cell, because then you're eventually going to pull them apart, right? To pull them apart, you need some microtubule disassembly. And you need to be where they're pulled opposite like this, which is what would occur during anaphase. Now, if you don't have microtubule disassembly, they're going to essentially get stuck and you can't progress. It's just not going to happen. And so if it gets stuck, you can't make it past this checkpoint. You're kind of stuck on this mitosis check, this M checkpoint here. And what happens is that actually can force the cell either into G0, or in this case usually is apoptosis, because it's stuck at the checkpoint. It's not moving forward. Other mechanisms of the cell detect this, and they say, hey, you know what? We got to just cut our losses here. There's something wrong. And so the cell goes into apoptosis, and that's essentially how these drugs kill cancer cells. And specifically, which drug does that is paclitaxel. So paclitaxel does this. It prevents mitotic spindle breakdown, which is really important. However, these two drugs, vinblastine and vincristine, they inhibit mitotic spindle formation. So that's more of what traditionally what you think. This is kind of an inverse way of doing it, preventing the breakdown. This is more of a direct mechanism, what you would think of. And so you remember you have those alpha and beta dimers that assemble to form, these microtubule. This is what it would look like on a micro level here. So these are assembling here. And so what these drugs do is they come in and they prevent this from happening. So why that's important during mitosis is that you need these microtubules to be binding to the chromosomes so that they can bind them and then help migrate them to the center of the cell during metaphase so that they can be separated. So again, that was just kind of a quick review of these drugs and how they act. But again, the take-home message is that these are acting in the metaphase portion and thus the M phase of the cell cycle. So over the next few slides, we're going to talk about chronic myelogenous leukemia, or as it's commonly known as CML. Now, this is actually a pretty rare disease, but it's a disease that comes up a lot in biochemistry, pathology, and even pharmacology, and you'll see why over the next couple slides. And this is a extremely high yield disease that you want to be aware of because it comes up in all three of those subjects. So therefore, you could see it on an exam in any one of those subjects. You could see it on a board exam for sure. And so you definitely want to know this disease well and know its treatment well. So it's a form of leukemia. So it's a blood cancer caused by chromosomal translocation. That's where this biochemistry part comes in, between parts of the 9th and 22nd chromosomes to then create the Philadelphia chromosome. And so it's called the Philadelphia chromosome because this was discovered by scientists in the city of Philadelphia, and they decided to name it after the city of Philadelphia. So you come down here and you have the ABL gene, or ABL, which is on the chromosome 9, and you have the BCR gene, which is on this 22nd chromosome. So then you have a translocation where you essentially just swap out part of a chromosome for another. And so you have this part of 22 that moved over here carrying the ABL gene, and now it's in proximity with the BCR gene that was on the 22nd. And then you have this part of 22nd that's moved on to the 9th. And so this is the convention of how you would write a translocation. So you have T9, 22. So this is just T for translocation, 9 and 22 for the two chromosomes for which the translocation occurred. Now what has resulted here is that you have what's called a fusion gene, where BCR and ABL genes have fused to create what's called a fusion gene. And this fusion gene codes for a unique protein, so not a protein that's normally produced in the cell. That's a receptor tyrosine kinase. So this is a cell surface receptor, the BCR-ABL protein. And what's unique about this is that it's continuously active. It doesn't require any external activation. So this is a perfect example of what we were talking about on the slide where we were talking about oncogenes. So this protein is created by this fusion that's a result of this translocation. And what happens is that since it's not regulated, this cell surface receptor, it stimulates a signaling pathway that drives unregulated replication of cells, specifically myeloid cells in the bone marrow. So myelogenous leukemia. Now, as far as the treatment goes, let me just digress here for a second, tell you a quick story about this. The drug imatinib, or known by its trade name as Gleevec, actually made the cover of Time magazine because CML before Gleevec was actually a fatal disease. It would kill people of all ages, people in their 20s and 30s, elderly people. There was no effective treatment for it. And what happened was they had these patients go on this clinical trial, phase 1 trial, where you're just really even just seeing it to make sure it doesn't kill people or cause horrible side effects. You put them on this drug, they put them on Gleevec, and it cured almost everybody in the trial. It was literally a miracle drug. It was a sensation, and it made the cover of Time magazine, and it was really a triumph for cancer therapy and especially targeted chemotherapy development. You know, I've talked a lot about the difference between cytotoxic chemotherapy and targeted chemotherapy, and made the cover of Time magazine. And they followed the patients up, and almost all of them were still in remission at 5 years after being treated. And so it's really just an amazing example of what medical research can produce. And so I thought I'd just share that because it's a nice story, and it can help you remember this as well. So anyways, why you really care is because it's on your exams and it's very high yield for your boards. And so what you have here is it's a targeted drug therapy, and it specifically inhibits the BCR-ABL protein. And that's why this drug was so effective, is because this protein is not normally present in normal cells. And so it really selectively targets the cancer cells, because this is only found in the cancer cell. It's the only cell in the whole body in these patients where this is found. And so it results in specifically CML cell death while sparing the healthy cells. And so as you can see here, it binds to it, renders it inactive, and the cells die off. And so it really, again, was such a great example of where targeted therapy can really work well, and why targeted therapy is worth pursuing, because not only can you have great results, but you can also hopefully spare healthy cells, which when you spare healthy cells, that helps decrease the side effects. Li-Fraumeni syndrome. This is a inherited mutation in the tumor suppressor p53. Again, p53, as we say here in the text, it monitors DNA damage, and it causes arrest of the cell cycle if there's a mutation found. Again, it's at this G1S checkpoint, and so it allows for damaged DNA to be repaired. If the damaged DNA is beyond repair, then it sends the cell into apoptosis, and again, that's important for preventing cancer. So as you can imagine, if you inherit a mutation in p53, devastating effects. And so again, just to review how it works, p53, especially for this checkpoint here, this G1S checkpoint, it induces transcription of these proteins, which then help with preventing phosphorylation of the RB protein, which then helps hold ETF, and thus preventing this and preventing this. And so it prevents progression of the G1 phase into the S phase. And so as we say here, it's thought of as the guardian of the genome, because it really guards this major league checkpoint before you enter DNA replication. And so this is an autosomal dominant inheritance, and it leads to early and varied cancers. And this is where you'll see children develop cancers that you normally wouldn't see until much later in life. And unfortunately, these children, they develop multiple malignancies, including breast cancer, colon cancer. They can develop blood cancers, leukemias. They can develop sarcomas. And so it's a devastating disease, and it all stems back to p53. And it really shows how important p53 is in normal cell cycle progression, and then in preventing the development of malignancy, because these children unfortunately are at risk for developing multiple types of cancers that are usually just not found until later in life. And so it's just a terrible disease. So we've already talked about the retinoblastoma protein, or the RB protein, as we show here in this diagram when we talked about cell cycle regulation. So we're going to talk about retinoblastoma, because studying retinoblastoma led to discovering how the RB protein functions. And sure, it's named after retinoblastoma, but the RB protein is it's involved in cell cycle regulation in all cells. And so it's important for not only retinoblastoma, but other cancers as well. So retinoblastoma results from a mutation in the RB tumor suppressor gene on chromosome 13. Just to review here, RB binds to E2F, which is a transcription factor that promotes transcription and progression of G1 phase into the S phase. And so when it's bound here, when this is active, this is inactive. And so you prevent this and you prevent this, and then it helps prevent unregulated cell proliferation. Now, retinoblastoma can be inherited or sporadic, and this is where what's called the two-hit hypothesis comes in, which is what we'll talk about in the next couple of slides. And this is a good example of showing how a proto-oncogene can then turn into an oncogene through mutations, and then an oncogene can develop into cancer. So the two-hit hypothesis states that both copies of a gene must be defected in order for someone to develop retinoblastoma. Now, we're going to talk about retinoblastoma, but this hypothesis was actually developed to apply to really all of cancer. both oncogenes and then tumor suppressor genes as well. And this can happen either through two random mutations that occur by chance, sporadic. So if you remember, you get two copies of one chromosome. So we'll just say chromosome 13 in this case, because that's for retinoblastoma. And you received one from mom and one from dad, right? It's the exact same chromosome. Remember, originally you received 23 from mom, 23 from dad. So you have two copies of the chromosomes. And so for this, chromosome 13, you need a mutation on both of these to develop retinoblastoma, okay? So what happens is you can either have both of these, and eventually over time you develop a mutation in one of them, and then eventually again you'll develop a second mutation on the other copy in the exact same cell. It's got to be in the same cell, because then you've got to replicate this, and that's how you develop cancer. So that's the sporadic. Now the inheritance is where you actually inherited one copy. It could be from mom or dad, where you already have a mutation. So the likelihood of getting to where both are mutated is much higher in these patients. And so they develop it much earlier. So this is the inherited, this is sporadic. You're essentially depending on two sporadic mutations occurring, hence the sporadic. Versus this one, you've already inherited one copy that's mutated and you're trying to get to two. So we have some figures in your book we'll go through. Sporadic, it usually occurs in adults because it just takes more time to get both copies in one cell mutated. And so because of that, it occurs in adults. And it's been found where you only have tumor development in one eye. So you have sporadic, both are normal, both copies are normal. Then eventually you acquire a mutation. And then again over time, you acquire another mutation. And so by acquiring that one mutation, you've increased the potential compared to normal chromosomes, which are these guys here. These are all normal. But however, since you've already developed this one mutation, you have an increased chance of another sporadic mutation, which will then cause tumor formation. So this cell with these chromosomes, with this mutation already occurring, are already at an increased chance compared to these ones that are still normal. Then you have the hereditary or inherited. So this usually occurs in younger adults and even children, because it's much easier and much more likely to develop tumors, because you've already started with one of the copies mutated, as you see here. Now, some patients, they can just live their lives and they can have where they're like these guys, where the cells only have one mutated copy. These do not develop into cancer because you need both, hence the two hit. You need two hits, two mutations. Now, if these develop into where you acquire another mutation on the other copy, it's got to be on both copies in the same cell, then you develop tumors, because then these cells are going to replicate, and then you have tumor formation. Now, this makes it much more likely to occur because you've already started out with one. That's why it happens in younger adults and children. And then they've found that it generates tumors in both eyes. And the other thing they've found is that these patients with the inherited version typically usually at more risk for developing other types of tumors, because since the RB protein is involved in cell cycle regulation for all cells, this is going to affect generating tumors in other aspects as well. And so then these patients can develop other types of tumors as well. Clinical features: retinoblastoma, it's eye cancer, and then they have leukocoria, which is loss of the corneal red reflex on eye exam. So pay attention if you see that in a question stem. And then these patients, especially the inherited version, they're at increased risk of developing osteosarcoma or bone cancer. So now we'll finish this up with Lynch syndrome and FAP, and we'll differentiate. So Lynch syndrome, this is a DNA mismatch repair genes, MLH1. This is a specific name of the genes. They are methylated. Now remember, when you methylate a gene, you essentially turn it off. So you have decreased expression. This actually leads to increased rates of mutations because you don't have these repair genes. If there's damage in the DNA, you can't repair it. So then it progresses past that stage of regulation, DNA replication. And this leads to what's called microsatellite instability. And this instability induces more risk of mutations occurring. So it's a domino effect, if you will. The inheritance for this is autosomal dominant. And the clinical features is that the disease progresses to colorectal cancer. However, it's at an older age than FAP. That's one way to differentiate it. These patients are going to be much younger. And also these patients will be at risk for developing endometrial and ovarian cancers in women, and then also skin cancer. And this is another thing that helps differentiate it from FAP, which we'll talk about in the next slide, because FAP is more exclusive to colon cancer. And so these patients, if they're developing these and then they're developing colon cancer at an older age, it's going to be more likely to be Lynch syndrome or hereditary non-polyposis colorectal cancer, HNPCC. So lastly here, FAP, familial adenomatous polyposis. This is a mutation in the APC tumor suppressor gene found on chromosome 5. This is actually found to follow that 2-hit hypothesis that we talked about with retinoblastoma. So again, in this case, you're going to inherit 2 chromosome 5s, one from mom, one from dad. They each have a APC gene on each one. However, you're going to inherit a mutation in one of those APC genes. The other one is going to be normal, could be from mom or dad. Then over time, what's going to happen is the other chromosome 5, so this one was already mutated, the APC gene on this one is then going to develop a mutation, and then that's where you're going to develop FAP. So this leads to increased rate of mutations via chromosome instability, which increases the risk of additional mutations. Same thing, that domino effect. This inheritance is autosomal dominant. Clinical features, it's going to lead to polyps, literally thousands of them, throughout the colon around the puberty age. So very, very young. Usually you don't start doing colonoscopies until around the age of 40. And as a result of this is going to lead to earlier occurrence of colorectal cancer than HNPCC. So that's one way you differentiate it. And the other thing is all of these patients will eventually develop colon cancer. Now, the treatment is that you remove the colon because they have all these polyps and they're going to develop into cancers. So you need to be proactive here and help prevent it. And that's actually through a surgery called a colectomy. All right, so that closes out our discussion of clinical pearls relevant to the cell cycle. And it also closes out our third and final lecture for Chapter 5, Cell Replication and Cell Cycle.